Okay, welcome to Screencast about photosynthesis. Wah! And the crowd goes wild. Okay, so I will go ahead and share my screen. All right. So I will say that I highly recommend that you watch Mr. Anderson's little movie on photosynthesis. I will post it, make sure it's there. And this will give you a nice overview of photosynthesis. So I strongly recommend that, but it's not going to be homework. So I will say this before I start photosynthesis is that in years past, because cellular respiration takes students a long time to grasp all the components, and I make you do that pamphlet, that students often spend a lot of time on that, and then they fail to really grasp photosynthesis. So I'm telling you that now as a warning that photosynthesis is just as important on the test as cellular respiration. So make sure that you are spending enough time on it as well. All right. So first thing first, hold on, let me grab my notes, is that you need to be able to obviously tell us or write down the equation for photosynthesis, all right? Which you should have a pretty good idea because it's just the reciprocal equation to, what's it called, to cellular respiration, right? Sorry, organizing the studio. All right, so for photosynthesis, I'm gonna just write it, I guess down here. So we need, whoa, that's a thick pen. So we need sunlight in, we need carbon dioxide in, and actually, I'm gonna write the balanced equation, 6 CO2 plus 6 waters, and that is gonna go to C6, oops, sorry, C6H12O6 plus our 6 oxygens, all right? So the question now is what was oxidized and what was reduced? And again, the way I always think that it's easiest to look at the carbons, right? So we're going from carbon dioxide to our glucose, and we added an H, right? So that was a reduction. And then we are going from water to oxygen, right? And that was going to be our oxidation. So many people, when they started looking at photosynthesis and cellular respiration, they assumed the gas became the gas, right? That CO2 turned into oxygen, but that is not the case, right? So that's just something to think about. So you should know what's oxidized and what's reduced. Remember, the way I think about it is if it was oxidized, oh, good, it lost that H, you put an X through it for an oxidation. All right. So the overall reaction, which we just wrote out previously, 6 CO2 plus 6 waters to sun. And we just talked about what was oxidized and what was reduced. So the thing to think about here is that photosynthesis occurs in two kind of independent but united mechanisms, right? So the first is the light well, not the first. One of them is the light independent reaction. That is going to take place in the stroma. That is where we make sugar. That's where we're going to make our glucose, right? So we're going to turn our carbon dioxide into glucose. And again, that is a reduction. We're adding. And then we have light-dependent reactions. Those are going to take place in the thylakoid membranes. In the thylakoid membranes, those little stacks of pancakes. And this is where we're going to break water, right? So in the light-dependent reactions, we break water up, and that is where we're going to kind of get water going to oxygen, and that is going to be our oxidation, right? Excuse me. And remember, this is just the reciprocal reaction to cellular respiration, where glucose was broken down or oxidized into carbon dioxide, right? So the gases do not become gases. So oxygen does not become carbon dioxide does not become oxygen. Rather, the carbon dioxide is always going to be oxidized or reduced in terms of the glucose. So always keep the carbons in mind. All right, so again, if we look at photosynthesis, we are looking at, and you've seen this before, this is going to be the buildup of glucose, remember? And if it's a buildup, it's going to be anabolic, right? We have an input of energy from the sun, making it endergonic, right? Because we're going we're putting an input of energy, that's going to be a positive delta G. And we're also going to be lowering entropy because we're building things up. Cellular respiration, on the other hand, is going to be breaking something down. So it's catabolic, right? We're going to get energy out in the form of ATP. So that is going to be exergonic. It's going to be spontaneous, meaning it's a highly negative delta G. And we are breaking things down. So we're going to see an increase in entropy, all right? So that was just a review. And that's maybe, hopefully you're seeing why I kind of forced you to know that early on. So you don't have to relearn these steps. All right. One thing to point out, it's very important to remember that plants do both of these things, right? They do both. So they have photosynthesis that occurs in their chloroplasts, right? And they have cellular respiration, which is going to occur in their mitochondria. Remember, all eukaryotic cells have mitochondria, right? So they're going to build up sugar in their chloroplasts. They're going to break it down in their mitochondria. And that allows plants not to have to eat their food because they make their own superpowers. All right, so the sun is our main source of energy. Photosynthesis is the process that's converting solar energy into chemical energy, right? So we're going to make bonds that are in the form of glucose, right? So it's really neat. We're literally converting energy from the sun into the energy in the bonds formed in glucose, right? In that way, directly or indirectly, photosynthesis nourishes basically almost the entire living world, right? So photosynthetic organisms feed themselves basically through the sun, right? They do that by the reaction above, sun to sugar. And so plants and algae both do that. There are some organisms called protists that are single-celled eukaryotic cells. And that would be, an example would be this guy. And that is a little euglena. And then we also have some prokaryotic cells, meaning some bacteria that can also be photosynthetic, right? So those are just some examples. Like here's, this one is algae, we have plants, we have single-celled, we have bacteria, etc. All right, so this brings us to what is the definition of those? All right, so autotrophs, that is a word you need to know, autotroph, those are the producers. The way I remember that, auto means self. I forget what troph means, but I always think of it as eat. So these would be self-eating. And so those, they produce their own. So they produce organic molecules from carbon dioxide and sunlight. So again, we're turning sunlight into the bonds of C6H12O6 into that chemical bonds. Whereas heterotrophs, so here, I'm gonna put this. So these are the plants, algae, protists, photosynthetic bacteria, right? Those are all autotrophs, or the producers of the world. Whereas most animal cells are heterotrophs, meaning, and the hetero means, other, many, different. I forget what exactly hetero means, but these are the eaters, right? So those are the consumers or the eaters. and they eat organic materials. So they either eat plants, right? Or they eat animals that eat plants, right? So plants really drive all of the energy on our planet, right? And they do that by harnessing the energy of the sun, right? So they harness the energy of the sun to make organic molecules, right? So that's what the autotrophs do. They harness and they make those. The heterotrophs will eat that, will break it down to provide carbon dioxide for the autotrophs, right? For the plants. Again, just want to point out is that energy in its disordered form, called heat, in its most with highest entropy, heat, and that is often lost whenever you have a reaction, right? You get a little bit lost as heat. All right, so solar energy being converted into chemical energy, right? And just a quick reminder of light and how light travels. It travels in these kind of waves, right? And the visible light spectrum, which is the only light that humans can see, that most animals can see, not all, are in this wavelength of 380 to 750 nanometers, right? You should know that shorter ones have higher energy, longer ones have lower energy, right? So what is really cool that never ceases to like blow my mind is that when light hits matter, it can do three different things. It can reflect off that material, it can be transmitted through that material, or it can be absorbed by that material, right? So what's really cool about plants, and I love this, is that the light that is reflected off is what goes into our eyes, right? And that is green, right? So green light, plants are green because that is the wavelength of light that gets reflected, right? So here, this is reflected off of plants, right? And that's why it's green. What's neat is that in plants, there are, let me go to green, well, actually, let me go to red. There are photopigments, right? Which kind of mean they're light capturing colors in plants that can absorb the light, right? So chlorophyll, you've learned before that chlorophyll is why plants are green, right? Chlorophyll itself doesn't absorb green, but it reflects green. And that's why we see that in our eyes, right? What it does is it absorbs blue and red, right? So this blue and red light, that is what plants absorb through chlorophyll. But they reflect the green back to our eyes, right? So chlorophyll absorbs red and blue light and reflects green. That's why plants appear green to us. And I know since it's fall, and I used to teach this on the East Coast when we have these beautiful fall colors, but we see a little bit here in San Diego. And I want to tell you about this other one called carotenoids. All right, so this is another photopigment, and these reflect red, orange, and yellow, right? And that's what makes the fall leaves, right? So during the growing season, chlorophyll is replaced constantly in leaves, right? And chlorophyll, but chlorophyll, however, breaks down with exposure to light in the same way like a colored paper does if you put it on your window for long periods of time. So the leaves must manufacture new chlorophyll to replace the chlorophyll that was lost, right? So in autumn, when the connection between the leaf and the rest of the plant begins to be blocked off, the production of chlorophyll slows and then stops. And within that very kind of short time, chlorophyll disappears. So chlorophyll, which was absorbing red, blue, reflecting green, breaks down, and now you get these carotenoids exposed, right? And the carotenoids now reflect all those beautiful fall colors. So that's why leaves turn color. How cool, right? All right, so photosynthesis occurs primarily in the leaves, and we've gone over this a bit, so I'm going to do it quickly. The leaves are green due to this chlorophyll reflecting the green light back out, right? So when we look, we see that green color. That's an eyeball, by the way. And if you look at a cross section, in plants, we have this kind of meaty section, and that's called the mesophyll cell. That's like the mesophyll of a leaf. In those mesophyll, you have many, many mesophyll cells. Each mesophyll cell has a bunch of chloroplasts, about 30 to 40 individual chloroplasts. And remember the structure of a chloroplast. We have an inner membrane and outer membrane. We have thylakoid membranes. We have stroma, and a stack of those thylakoids is a granum or many grana. So light is absorbed. What we're going to see is that light is going to be absorbed by chlorophyll, and that's going to happen on the thylakoid membranes. And that is what's going to try to harness that solar energy and help convert it to chemical energy, right? So chloroplasts really are solar-powered chemical factories where they take solar power in and they create chemical power out, chemical energy. Oh, so I want to point out, if you look here at the bottom of a leaf, you can see that we have what are called stomata, all right? Stomata. It's one of my favorite words in biology. I've got a couple of them, but that's one of them. It's called stomata. And this is where plants are going to do oxygen exchange. They're going to take carbon dioxide in and oxygen out, right? And some people did this on their cell project, so you may remember that. So stomata on the underside of leaves allows for gas exchange. They're literally openings on the underside of leaves that help carbon dioxide go out and oxygen go in. And I'm going to share a quick little movie if I can get it together. Hold on, let me. Okay, let me share this very quick movie to show stomata under a microscope, and you can actually see them opening and closing, right? That's jazzy music. So here we can see them open, close. Just pay attention to one. Ready? Open, close. Now let's look at this one. Open, close. Open, close. So you can actually look at that. And this was, stop that sound. So this was on a 400x microscope. The shots were captured once every 100 minutes, right? And then they compiled them into 10-second long movie, just to give you a sense. So it's not real time. They're actually slowed down quite a bit. Okay, so let me stop sharing and I'll share my other screen. Okay, so stomata are on the underside of leaves. And we're going to see that plants take carbon dioxide in, right? And they give off oxygen as waste, which is perfect because animal cells love to use that oxygen for cellular respiration, right? So if you look at the overview, again, of photosynthesis, I just want to remind you that we have these two different reactions. One are the light independent reactions, where we are going to be converting carbon dioxide to glucose. And then we'll have the light dependent reactions, where we're going to split water, break up water, and to create our oxygen, right? I don't care that you know about this complex reaction, but I do care about that you understand this balanced equation. All right, so photosynthesis occurs in two phases, the light dependent reactions and the light independent reactions. And the light independent, or the light dependent reactions, this is the photo part, light dependent, photo, right? That's the capturing of sunlight. The light independent reactions are the synthesis right? That's going to be where you make your glucose. So if we look at this, we have light-dependent reactions. These need sunlight, right? This occurs in the thylakoid membranes, right? So on those little stacks, we are going to see that we're going to break water up and we're going to release oxygen out, right? This light dependent reactions, we're taking sun in and we are making energy with it. In the form of ATP and NADPH, right? So remember in cellular respiration, we had NADH and FADH2. In photosynthesis, we have NADPH, but it's easy to remember because photosynthesis and we have that pH, right? The light independent reactions, no sunlight needed. This happens in the stroma, kind of the fluid of the cytoplasm, or sorry, of the chloroplast. And here we are going to take CO2 in, and we're going to see how that harnesses, how we can convert that into an organic molecule, right? We are going to use the energy made from the light dependent reactions, and we are going to convert them. We're going to use them and give off ADP and NAD plus, right? So we're going to break these down and they're going to be converted back, right? So if you look at the overview, I'm going to clear the page here. So this is a really important image that you really need to fully understand and be able to draw and know, right? We're going to get into a lot more nitty-gritty details, but this is essential. So in the light dependent reactions, what we're going to see is that water comes in, right? And we're going to see that comes from the roots, right, up the xylem. So water comes in. In the light dependent reactions, we're going to see that sunlight comes in and we're going to break water up, right? We're going to see a lot of nitty-gritty details in there. But what we're going to have is that oxygen is released, right? That energy from the light dependent reactions is going to create ATP and NADPH, right? Those are absolutely required for the light independent reactions, which take carbon dioxide in to create sugar going out. They're using the energy in ATP and NADPH, and they're going to recycle the products back after that, right? So in the light dependent reactions, water in, oxygen out. In the light independent reactions, CO2 in, and we're going to see that that makes our glucose out. And that glucose can then be used directly for cellular respiration in the plant's mitochondria. Okay, so I'm going to skip the drawing, but I'm going to go over this, right? So again, we have, whoops, this is going to be H2O. So I'm going to, whatever, this is literally the diagram that we just had, where we have water coming in, oxygen coming out. We are going to use the light dependent reactions to create energy in the form of ATP and NADPH, right? Those are used to power the Calvin cycle, where we're taking carbon dioxide in. When we take carbon dioxide in, we're going to get sugar out, right? So this energy from the light dependent reactions is used to power the formation of glucose, and we're going to recycle these byproducts back to the light dependent reactions so that they can make more ATP and NADPH, right? Okay, so the light dependent reactions occur in two photosystems, right? And I'm going to go through, I'm going to have a video where I draw these out, so don't worry. But the photosystems occur in the thylakoid membranes. And a photosystem, I get over and over again, what is a photosystem, all right? A photosystem, all it is a group of proteins and pigments, right? So proteins and chlorophyll, primarily. Remember, chlorophyll absorbs blue and red light and reflects green light. And unfortunately, because science is annoying, photosystem 1 was discovered first, but it comes second. Oh, so annoying, right? So what happens, and the reason that photosystem one, well, whatever. Okay, so we're gonna just go through it in order. So first thing is that we have photosystem 2, right? So in photosystem 2, we're gonna get light coming in, right? Light comes into photosystem 2. We are gonna see that water splits, right? And this is not due to the light coming in, but in my mind, that is the easiest way to remember. Light comes in, breaks water, right? When water breaks, just like the opposite of cellular respiration, we're going to get 2 protons out. We're going to get half an oxygen, or just 1 oxygen atom. And really importantly, we get 2 electrons, right? Those 2 electrons get powered by that sunlight and by that photosystem, and they get energized. And they get energized to electron transport chain, right? So in this electron transport chain, we know what happens in electron transport chains, right? Electrons get kind of shuttled around. When they do that, they are pumping hydrogens, right? Here, we're going to pump those hydrogens into the thylakoid membranes. Those H's are going to go down their concentration gradient through ATP synthase to create ATP, right? That should all sound pretty familiar to you, right? Then we have photosystem 2, so photosystem 1, right? Here we have light energy coming in again, and we get those electrons being energized, same electrons from the splitting of water, right? They get passed from electron transport chain in photosystem 1 to photosystem 2. We have another ETC, electron transport chain. And here, instead of driving ATP synthesis, we are going to make NADPH, right? And here we have NAD plus as the final electron acceptor. What was the final electron acceptor in cellular respiration? That was oxygen. The final electron acceptor in photosynthesis is NADP plus, which will make NADPH, right? So the light-dependent reactions split water. The electrons go into the electron transport chain. Oxygen goes out as waste. And what we're going to see is the electrons go through the first electron transport chain, makes ATP. Second electron transport chain makes NADPH. So in a very, very kind of reduced way of thinking about it, the light-dependent reactions kind of are like a pinball game where we have photosystem 2. We get a photon of, we have a photon of light coming in. It energizes these electrons that come from the splitting of water. They energize it up here, and then we have an ETC, right? So the ETC will make ATP. Then we have another photon of light coming in. It'll energize those electrons. Those electrons are going to go down an electron transport chain to generate NADPH. So here's a kind of a more complicated version. I just want to remind you that we are dealing with the light-dependent reactions, where we are using sun to create ATP and NADPH, right? We're going to get water splitting. We're going to get electrons going into the system, and we're going to get oxygen out of the system, right? So if you look at this diagram, we are really on the y-axis here looking at the energy level of electrons. So at the very beginning, we're going to see that water splits, right? During that splitting, that gives us electrons that can go into the photosystems. We are going to see, oxygen that is going to go out as waste, right? And then we have some hydrogens here, which can be used over here, right? So everything is kind of accounted for. So light comes in, again, to photosystem 2, right? Those electrons from water get energized. They will go down the electron transport chain, which will make a gradient in the thylakoid membranes, which will be used to drive the synthesis of ATP. We get another photon of light in. Those same electrons are going to get energized, go down another electron transport chain to the final electron acceptor, which is NADP plus, to make NADPH. All right, so here's a model from one of the Pogels, which I think is like, for me, the easiest way to look at this. All right, so we have, so I'm going to trace the electrons through, right? So we have light coming into photosystem 2. Water is going to get split. Those electrons from water are going to get energized. They're going to go through the electron transport chain, right? As you go through electron transport chain, you are going to be pumping these H's into the thylakoid membrane to create, as we know, an electrochemical gradient. Right? As you build up those H's, they are going to flow down their concentration gradient through our friend ATP synthase, right? And we know that as you do ATP synthase, you convert ADP and PI into ATP, right? You're going to make lots of ATP. Those electrons, though, are not lost. They're still going through the ETC. And then once they get to photosystem 1, another photon of light will energize them. And they're going to go through another ETC until they reach their final electron acceptor, which is NADP+, final electron acceptor. And that is going to then make our another output, which is NADP+, right? Because I like this one because you can really see how the electrochemical gradient is building up inside the thylakoids, right? ATP is being made in the stroma along with NADP+. So here's just another view of that. So you can take whichever one you like the most. But again, light energy comes in, water was split, the electrons go into photosystem 1, they get energized, they're going to go down the ETC. This is electron transport chain 2 because it's photosystem 2, right? They're going to go down these electrons. As they go down the ETC 2, you're going to see hydrogens being pumped into the thylakoid space, right? Inside the thylakoids. That gradient, these hydrogens can flow down their concentration gradient through ATP synthase to make ATP. These electrons, in the meantime, are going to continue through to photosystem 1, where they get re-energized by light. They're going to go down ET, electron transport chain 1, because it's photosystem 1, until they reach their final electron acceptor, which is NADP+, to create NADPH. So it's really important that NADPH and ATP have been made in the stroma, because those are going to be used to power these light independent reactions, which are going to occur in the stroma, right? So you should be looking at the similarities to cellular respiration and the differences. And in particular, you should focus on the location and types of cells involved. What are the electron carriers, right? So we have NADH and FADH2 versus NADPH, for example. The source of electrons, where do the electrons come in photosynthesis versus cellular respiration, right? In photosynthesis, they come from water, but think about where they come from in cellular respiration, right? What are the final electron acceptors? Oxygen versus this NADP+. Where are the location of the gradients that build up, right? Thylakoid membranes versus intermembrane space. And think about the inputs and outputs. Making a chart of this would be really useful. All right. So again, chemiosmosis in mitochondria versus chloroplasts. So we've done this before, but we're going to go through it again briefly. So in the mitochondria, the H's are pumped from the electron transport chain into this intermembrane space, right? And if we look here we have proteins that are on these cristae, or the cristae, the intermembrane, and we have a little ATP synthase, right? So electrons from NADH and FADH2 are kind of transferred. In the meantime, you are going to be pumping H's in. They're going to flow down their concentration gradient to make ATP in the matrix. In chloroplasts, on the other hand, we see that in the thylakoid membranes, that's where we have these important proteins and photosystems. And we're going to see that the H's are going to build up in the thylakoid membranes. We are going to have ATP synthase here, where the hydrogens are going to flow down to make ATP in the stroma. All right. So we have now covered the light-dependent reactions, and now we're going to cover the light-independent reactions, which are called the Calvin cycle. And Calvin cycle, I always remember, because Calvin cycle equals carbon, right? We are dealing with converting CO2 into C6H12O6. So the light-independent reactions occur in the stroma, and they're going to be powered by ATP and NADPH that we got from the light-dependent reactions, right? So the glucose that we're going to get out in the Calvin cycle they can be used for starch. Remember, plant storage. They can also be used for cellular respiration to break down that glucose for energy. Okay, so where do the light-independent reactions take place? That's going to be in the stroma. And just looking at this diagram, without me telling you anything, you should be able to answer these questions, right? So this is kind of what the Pogles do. They give you something where you have all the information, but you really need to dissect those figures to figure out what's going on. So describe what carbon fixation is, right? So carbon fixation is basically taking carbon dioxide from the air and turning it into a usable organic molecule. And if you're thinking, yes, of course we know that carbon dioxide turns into glucose, but the carbon fixation phase happens earlier than glucose, right? So we're taking carbon dioxide and we're creating this molecule. You don't have to know the names of all of these, but we're going to create a molecule that is an organic molecule, right? So carbon fixation is that first phase, right? Locate the following molecules and answer the questions below. So how many carbons are in RUBP? So we're going to look for RUBP. That's here. We'll just count the carbons. So 1, 2, 3, 4, 5 carbons. So RUBP is a 5-carbon molecule. You're going to see that these 5 carbons these 5 carbons are going to be added to the 1 carbon to make 6 carbons. Those 6 carbons are then going to be split into 2 3 carbons, which should sound familiar, right? Just like glucose. Okay, so how many molecules, how many carbons are in PGA and G3P? And again, I'm not going to make you memorize all of these crazy things, but you should be able to look at diagrams and figure them out. So PGA is a 3-carbon molecule, and G3P is also a 3-carbon molecule, right? So we have added a 5-carbon plus a 1 carbon to get 6 carbons, which then split into 2 3 carbons. Those 3 carbons get converted to a different 3 carbons. And we're going to see that, a different 3 carbons that are going to be used to make those things come together to make glucose, right? So how many molecules of PGA and G3P come from one RUBP? So one RUBP, right, is going to give us 2 G3P and 2 PGAs. So where does ATP and NADPH originate from, right? We know that ATP and NADPH came from the light-dependent reactions in the stroma, right? And you can see here that we are going to use ATP in multiple steps here of the Calvin cycle, and we're going to use NADPH. We're going to use that energy harnessed from the sun in order to do this cycle, right? How many turns of the cycle to make one glucose, right? And it turned, just a minute. Oops, sorry, hold on. Oh my God, my son is playing the recorder and it's really precious and really loud. So just a moment here. All right. So I'll show you an animation of this to show how we go here. All right. So in order to make glucose, we're actually going to have to see this cycle turn twice, right? So we're going to have to see this cycle 2 times to make one glucose molecule. And I'll show you an animation of that as well. Okay, so there's 3 main steps in carbon fixate, or sorry, in the Calvin cycle of these light independent reactions, all right? And there's a good animation here that you can look up. So number 1 is carbon fixation. Again, making CO2 from the air usable. So we're gonna add 5 carbon molecule plus a 1 carbon dioxide. And we're gonna fix that into 6 carbons, 1, 2, 3, 4, 5, 6. They get quickly broken into 2, 3 carbon molecules, right? The second phase of the Calvin cycle is to create G3P, right? So G3P is what's going to be used to power glucose production, right? And the reason it takes 2 turns of the cycle is that during 1 turn, this G3P, remember we had 2 of them, 1 is going to be recycled into the cycle and 1 is going to be put out of the cycle. So you have to go 2 times in order to get 2 of these molecules that can come together to make glucose, right? So 1, carbon fixation, 2, generation of G3P, and then 3, the regeneration of RUBP, right? That 5 carbon molecule. I promise we're almost done. So the G3P is called glutaraldehyde 3-phosphate. I don't care if you know that name at all, but what you should know is that G3P is a 3 carbon molecule that is the raw materials in order to make our glucose, right? And the Khan Academy does a really beautiful job looking at the Calvin cycle. I think this is an image from them. So for example, let me see here. You should be able to answer these kinds of questions. So how many turns of the Calvin cycle to make 1 G3P coming out, right? And that would be 1 turn. How many carbons are in a G3P? We should know that's a 3 carbon. How many carbons in glucose? You know that that is 6 carbons, right? How many G3Ps are needed to make 1 glucose? You need 2 3 carbon molecules to make glucose. And so how many turns to make 1 glucose? So 1 turn, we get 1 of these out. The other part gets recycled. So we need 2 turns to make another G3P, which will be used, powered to make glucose. All right. So in order to make a single G3P, we're going to use a lot. And I don't care if you know the numbers for this. This is not as important that you know the numbers. But what you should know is it takes a buttload, right? So to make a single a single one of these G3Ps coming out, you need 9 ATP and 6 NADPH, right? So to make a single glucose, you need 2 turns of that cycle. So you need a lot of ATP, 18 ATP and 12 NADPH. You need a lot of energy. Remember, the only way you make these is from the light-dependent reactions. Okay, and we are going to stop there. All right, so I know photosynthesis is a lot, but again, students often don't study as much as cellular respiration, and that's a mistake because it's basically equal weighting on the exam. All right, chip, chip, cheerio, toodles, poodles.